Dear all, welcome to my channel Biology Notes. Today I will discuss about the glycogenolysis process through which glucose molecules are released into blood circulation from glycogen. Contents of the video include general overview, location of glycogenolysis pathway, steps of glycogenolysis process. Glycogenolysis is the biochemical pathway in which glycogen breaks down into glucose molecules. We all know glucose is a source of energy for the entire human body. During the fasting state, to maintain normal blood glucose levels, the liver plays a central role in producing glucose via glycogenolysis. Including liver, glycogenolysis also takes place in kidney and muscle cells. You remember one thing that muscle cannot release glucose directly to the circulation. The reason I will discuss later. So, the glycogenolysis pathway mainly takes place in the cells of liver, kidney, muscles, etc. Now, I will discuss about the detailed mechanism of glycogenolysis process through this diagram. Suppose this is a glycogen molecule. These are the glucose residues bind via alpha 1,4 glycosidic bonds. This is the branch of glycogen molecule. Here also, these glucose residues which are in alpha 1,4 glycosidic linkages, and this single glucose residue is with an alpha 1,6 glycosidic bond. So, in the first step, glycogen phosphorylase catalyzes the phosphorylasis of glycogen. Glycogen phosphorylase actually ruptures alpha 1,4 glycosidic bonds between the glucose molecules. Phosphorylase acts on the non-reducing ends of glycogen branches and releases glucose 1-phosphate. The repeated action of phosphorylase on successive alpha 1,4 bonds in a branch of the glycogen molecule progressively shortens that branch. But the enzyme cannot cleave alpha 1,6 glycosidic bonds at the branching points of glycogen chains. So, the enzyme stops acting on a branch when only 4 glucose residues are left. At this point, another enzyme, oligo alpha 1,4 glucan transferase, separates a trisaccharide, means these 3 glucose residues from this terminal branch, and transfers it to the end of a neighboring branch. Here it is attached via alpha 1,4 bond. So, this branch has only a single glucose with an alpha 1,6 bond. Now, amylo 1,6 glucosidase or debranching enzyme hydrolyzes the alpha 1,6 glycosidic bond and release that molecule as a free glucose. So, this same process will happen repeatedly on the branches of glycogen molecules. At the end, about 90% of glucose residues will release as glucose 1-phosphate and remaining 10% glucose residues will release as glucose molecules itself. Phosphoglucomutase next isomerizes glucose 1-phosphate to glucose 6-phosphate. In liver and in kidney, the cell membrane enzyme known as glucose 6-phosphatase hydrolyzes glucose 6-phosphate into glucose and inorganic phosphate. Then the glucose is released into the blood circulation. But muscle cells lack glucose 6-phosphatase enzyme. So, muscle cells cannot convert glucose 6-phosphate to glucose. As a result, muscle cells cannot release glucose to the circulation. So, in muscles, glucose 6 phosphate directly enters the glycolysis process. So, this is all about the glycogenolysis process. If you have any query, please write in the comment section. Like and share the video. Subscribe the channel and press the bell icon to get notification of new video. Stay with us, keep watching. Thank you.